Ever been stuck in like a crowded hallway?
Yeah.
Everyone's going one way, but you absolutely need to go the other way.
Ugh. Yeah, I hate when that
All right,
happens.
it's the worst. Well, it turns out that's a challenge that cells in our bodies have to deal with constantly.
Oh, really?
Yeah. Yeah, they're navigating this super complex microscopic world, and sometimes they even have to move against the flow of fluids.
Against the flow.
Yeah, especially in like the really tight spaces in our tissues.
Huh. I never really thought about it like that.
Right. It's pretty amazing when you think about it. So why would they do that? It seems counterintuitive. You'd think they'd just go with the flow, conserve energy.
Yeah, I mean, that makes sense from an energy standpoint.
Right. But it turns out that this upstream movement, it's actually really important for a lot of different things in our bodies.
Mm-hmm.
And it might even play a role in how cancer spreads.
Whoa. Okay, so how do scientists study something like that? It's got to be tough, right? Like trying to track a single ant in an anthill.
That's a great analogy.
Mm-hmm.
And you're right, it's not easy. But scientists have developed these really cool tools called microfluidic devices.
Microfluidic devices.
Yeah, they're basically like tiny obstacle courses for cells. They mimic the really cramped conditions inside our bodies so that researchers can see how different types of cells, like cancer cells too, navigate these environments with different flow conditions.
So they're like creating tiny rivers and streams for the cells to swim in.
Exactly. And what they've learned is pretty surprising. One of the biggest findings is that being in a tight space actually triggers this upstream movement.
Really?
Yeah. When cells have a lot of room to move around, they're more likely to just go with the flow.
Yeah.
But squeeze them into a tight spot and they're like, nope, I'm going my own way.
Interesting. So it's not just the force of the current itself.
It's more than that.
Yeah.
There's something about the confinement that makes them want to push back.
So like they're using the walls to kind of brace themselves?
Exactly. And get this, they can actually swim upstream even against some pretty strong currents.
Wow, that's impressive for something so small. What else is going on? What other factors play a role?
Well, sometimes cells catch a whiff of something tempting downstream. Like a chemical signal that's drawing them in.
Like a breadcrumb trail.
Exactly. And if that signal is strong enough, they might decide to go with the flow, even if they're in a tight space.
So it's like they're weighing their options, deciding if the reward is worth the effort of swimming upstream.
Yeah. It's a really complex decision-making process for such tiny organisms. But it gets even more interesting.
How so?
Well, cells also have this thing called mechanosensitivity.
Mechanosensitivity.
Yeah. It means they can sense and respond to mechanical forces. It's kind of like how you feel the difference between walking on a sandy beach versus like a rocky path. So they can feel their surroundings. In a way, yeah.
Yeah.
And some cells are much more sensitive to these forces than others. And that can affect whether they decide to fight the current or just go with it.
Okay, so we've got these external factors like the flow and chemical signals, and then the cells themselves have different sensitivities. So what's happening inside the cell when it actually makes that decision to reverse direction? How does it physically move upstream?
Well, it's kind of like a cellular dance party. The whole internal structure of the cell gets remodeled when it decides to switch directions.
Remodeled? Like how?
It's cytoskeleton, the network of proteins that gives the cell its shape and allows it to move. It basically flips around.
The whole thing?
Yeah, it's pretty amazing.
Yeah.
And the proteins that are responsible for movement, actin and myosin, they shift to this new front of the cell.
So they're rearranging to face the new direction.
Exactly, like a team of movers rearranging furniture to face a new door.
I like that analogy. What about calcium and NHE1? Do they play a role in this remodeling?
Definitely. Calcium acts like an internal alarm system telling the cell that things are changing. And NAG1, that ion transporter, it helps the cell swell a little bit, giving it a bit more leverage to push against the flow.
Oh, I see. So it's like inflating a tiny life raft to help navigate those rapids. But we've been talking about all of this happening in a super tiny space. What about the nucleus? Does it have any part to play in this whole process?
For a long time, we thought the nucleus was just a storage container for DNA, like a library.
Right, that's what I learned in school.
Me too. But it turns out it's so much more dynamic than that. The nucleus actually seems to be really important for how the cell senses and responds to all these forces.
Really? So it's not just passively sitting there?
Nope, not at all. It's more like the captain of the ship constantly monitoring the environment and adjusting course.
Okay, so the nucleus is calling the shots. That's pretty wild. How does it even do that? So the nucleus, it's connected to the cell's cytoskeleton?
The cytoskeleton?
Yeah, remember that network of proteins we talked about that give the cell its shape and help it move?
Oh, right.
Well, they're connected through this structure called the LINC complex.
The LINC complex.
Yeah, and this connection, it means that any forces that are acting on the cell, they get transmitted to the nucleus.
Oh, like a tug on a fishing line.
Exactly. And the rigidity of the nucleus, it actually makes a big difference in how the cell responds to these forces.
So a more rigid nucleus would be better at resisting the flow.
It seems that way, yeah. It's largely determined by this protein called lamin AC.
Lamin AC.
Yeah, it's like having a sturdy anchor versus a flimsy one makes a big difference in how the cell holds up against the current.
So we've been talking a lot about cells that are fighting the flow. But what about the ones that go with the current? Are they just passive passengers? Or is there something more to their story?
Oh, it's definitely not a one-size-fits-all situation. Even in those tight spaces where you'd expect most cells to be battling the current, some still choose to go downstream.
And why would they do that?
Well, it turns out that this downstream movement, especially in cancer cells, it might be linked to how cells communicate with each other.
Cell communication.
Yeah. They release these tiny little chemical signals that act like homing beacons. They attract other cells of the same type.
So it's like a cellular invitation to a party.
Exactly. And when there's a current, these signals can get swept downstream, creating a trail that other cells can follow.
So even though they might be facing resistance, some cells are actually drawn in by this chemical trail.
Yeah, it's this fascinating example of how cells have to weigh all these different factors. You know, the physical forces, the chemical cues to decide where to go. It's like a constant push and pull.
A microscopic dance of survival.
Exactly.
So it's not just about the external forces, it's also about the cell's own internal state and how it's processing all of these signals.
Right. The cell's shape and its internal structure play a huge role in its migratory behavior. Some cells, when they reverse direction, they undergo a dramatic shape change.
Like they're morphing into a more streamlined form.
Yeah, optimizing their ability to fight the current.
It's like they're changing their outfit for the big swim upstream.
Exactly. But the cells that are moving downstream in these tight spaces, sometimes they actually maintain their shape.
Even as they're being pushed along.
It's like they're rolling with it, adapting to the forces rather than actively fighting them.
So maybe their internal structure's different too.
Yeah, the way they remodel their cytoskeleton could be a key factor here.
The cytoskeleton again. Let's break that down a little bit more. We talk about actin and myosin shifting to the front of the cell when it's moving upstream.
Right.
So what's the breakdown for each protein?
Okay, so picture those movers rearranging furniture again. In cells that are moving upstream, you see actin, this key component of the cytoskeleton. It quickly shifts to the new leading edge of the cell.
Okay, so it's like the movers focusing all their effort on the new direction.
Exactly. But here's where it gets even more interesting. Instead of joining actin at the new front, myosin actually accumulates at the back of the cell.
At the back, on the downstream side.
Yeah.
So instead of helping to pull the cell upstream, it's more like it's providing a push from behind.
That's a great way to put it. It seems like myosin's role in upstream migration is about providing that extra contractile force, helping the cell detach from its previous anchor points and propel itself forward.
Okay, so it's like this coordinated push and pull system, actin leading the way, myosin giving that extra boost from behind.
Right, and this coordination is crucial for overcoming the resistance of the flow and successfully migrating upstream. But let's not forget about those signaling pathways we were talking about earlier. What's actually telling the cell to go against the flow or ride the current?
Yeah, what's the signal that's triggering all of this?
So it turns out that calcium, remember that multi-talented signaling molecule we talked about? It plays a key role here too.
Calcium's back.
When cells encounter these flow forces, their internal calcium levels rise. And this seems to trigger a cascade of events that leads to either upstream or downstream migration.
So calcium is like the conductor of the orchestra directing the cell's response.
Exactly. And one of its key targets is NHE1. Remember that ion transporter that helps cells swell?
Oh, right.
If you inhibit NHE1, it actually reduces the number of cells moving upstream, which suggests that it's important for providing that extra leverage to push against the flow.
So if you block the cell's ability to swell, you're essentially taking away its power to resist the current.
Yeah, that's the idea. But calcium's not the only player in this cellular drama. There's another key factor, and it might surprise you.
Okay, I'm intrigued. Lay it on me.
It's the nucleus again. Remember how we talked about its rigidity being important?
Yeah.
Well, it turns out that that's also crucial for the cell's ability to sense and respond to these flow forces.
Wait, so the nucleus is like a sensory organ too. That's incredible. How does that work?
So remember that LINC complex? The one that connects the nucleus to the cytoskeleton?
Yeah.
That connection, it allows forces that are acting on the cell to be transmitted to the nucleus. It's like a direct line of communication, letting the nucleus know what's happening outside.
So the nucleus isn't just passively receiving information, it's actively using it to guide the cell's response.
Exactly. And the rigidity of the nucleus, it seems to be essential for this process. Cells with less rigid nuclei, often due to lower levels of that protein lamin AC, they're less sensitive to these flow forces and more likely to be swept downstream.
So it's like they've lost their grip, their ability to anchor themselves against the current. This makes me think about cancer cells again.
You're on the right track. We know that tumors often have really high internal pressure, which creates an outward flow of fluid. And as we've discussed, cells that are less sensitive to forces are more likely to be carried away by this flow.
So could this explain how cancer cells spread throughout the body? If they're less sensitive to those internal forces, it's easier for them to escape the tumor and travel to other areas?
That's the hypothesis. And it's actually supported by studies that show that highly metastatic breast cancer cells often have lower levels of lamin AC, and they're more likely to move downstream in response to flow.
Wow, this is also interconnected. So by understanding how the nucleus influences a cell's sensitivity to forces, we might be able to develop new ways to prevent cancer metastasis.
That's the hope. There's still a lot we don't know, but this research opens up some really exciting new possibilities for treatment, and it highlights how much we can learn by studying even the tiniest, seemingly insignificant details of cell behavior.
It's like those tiny cells are holding the secrets to some of the biggest challenges in medicine. This has been a truly mind-blowing conversation. I
Yeah.
feel like I've gone through a cellular boot camp.
It's a constantly evolving field, and I'm just as excited as you are to see what we uncover next.
Well, before we wrap things up, I have one final big picture question. If we understand all these forces and signals so well now, could we actually, steer cells to where we want them to go?
Like directing them to repair an injury?
Yeah. Or deliver a drug to a specific location?
That's a fascinating question, and it's something scientists are definitely working on. There are some really incredible techniques being developed to manipulate how cells move.
For real? Like what?
Well, for instance, researchers are using light to control how cells contract.
Wait, hold on. You're telling me we can control cells with light?
It's a real thing.
That sounds like something straight out of Star Trek.
It might sound futuristic, but it's a field called optogenetics.
Optogenetics?
Yeah. Researchers have figured out how to engineer cells so that they express these proteins that respond to light. And when these cells are exposed to specific wavelengths of light, it triggers all sorts of responses, including changes in their ability to contract.
Okay, so walk me through this.
Yeah.
They can basically shine a light on a cell and make it tighten up or relax.
Yeah, exactly.
It's insane. How do they even use that to control movement?
Well, by controlling those contractions, they can actually guide the cell's direction.
So they can steer it like a tiny little car.
In a way, yeah. It's pretty incredible. And think about the possibilities. Regenerative medicine, targeted drug delivery, it's mind-blowing.
It's amazing to think about how much we've learned about cells even just in the last few years.
Yeah.
And it's even more exciting to imagine where this research could lead us in the future.
Absolutely. This is just the beginning. Cell migration is this incredibly complex process with so many layers that we haven't even begun to understand. But with each new discovery, we gain a better understanding of the most fundamental processes of life.
It's true. It's like these tiny cells are holding the keys to unlocking some of the biggest mysteries in biology and medicine, too.
And sometimes the smallest things can reveal the biggest secrets, right?
That's a great way to put it. Well, this has been an incredible journey into the hidden world of cells. Thank you so much for taking the time to chat with us today.
It's been my pleasure. Always happy to talk about cells.
And to all our listeners out there, thanks for joining us on this deep dive. We'll catch you next time.